Hi everyone, my name is Mohammed Muzamil Khan. I am a PhD candidate in the bioinformatics program at Boston University. So today I'll be talking about my project Characterization and Integration of Transcriptional and Microbial Profiles of Oral Lesions in Cancer. So to start off with, head and neck cancer HNSCC is a devastating disease that arises in the upper aerodigestive tract and predominantly occurs in the oral cavity. There are two types of oral cancers, HPV positive, which could occur from human papilloma virus, or it could be HPV negative caused by extrinsic factors such as smoking, alcohol consumption, etc., or it could occur naturally. HPV negative cancers have a survival rate of nearly 65% during advanced stage and becomes less than 50% when metastasized. The oral cavity also consists of the microbes called the oral microbiota, which is the second largest after the gut and has shown to be associated with many diseases, including oral cancer. So it becomes important to identify these early drivers of malignant transformation for better diagnosis. So the aim of the study is to uncover these early drivers of transformation of HPV negative oral lesions and cancer compared to their profiles with the full-blown tumors and the baseline control group. So the oral malignant transformation goes through malignant stages, which occurs in a multi-step process where the tightly packed oral mucosa undergoes an aberrant change which enters into a hyperplastic state that moves on to a more progressed stage such as dysplasia and then carcinoma in situ, and then finally a carcinoma stage. A special case of hyperplasia called HKNR is being considered in this study that occurs naturally rather than rubbing of sharp tooth or braces, which is a common trait of oral lesions. The HKNR along with dysplasia is considered as pre-malignant lesions in this study. So our colleagues have already shown that HKNR and dysplastic samples show very similar mutational profiles. Like for example here, when you consider the top mutated genes in TCGA head and neck cancer and see the mutational profile of HKNR dysplasia, you see that they are very similar. So with this study, we are trying to use transcriptomics to help us uncover some of these differences, which will help us identify the differences between HKNR and dysplasia, and also help us identify important mechanisms of early transformation and support the design of novel interception strategies. So we generated a total transcriptomic sequencing profiles from biopsy samples of 66 patients comprising of healthy controls, the HKNR group and dysplasia group, which consists of more than 50% of the samples in the study identified as PMLs, and then we also have oral cancer. The mean age of all the patients was around 65 years, and an equal proportion of male and female is being considered in this study. The smoking status along with the progression status for some of these patients is being obtained, and that is also being considered for the downstream analysis. So to start off with looking at enrichment patterns of certain signatures that is relevant from other studies, we can apply GSVA enrichment algorithm that gives us an aggregate score that help us identify the enrichment patterns of certain groups when a reference compendium is being applied. So for this, we obtained signatures from 3 different studies such as oral premalignant disorders, the TCGA head and neck cancer dataset, as well as partial EMT profiles of certain head and neck cancers. So for bidirectional profiles, for example, for OPMD and TCGA head and neck cancer, we obtain the difference of up and down regulated GSVA scores, and the difference is being plotted here as a box plot. So of notice, what you see is, for example, for OPMD profiles applied on our data set, it shows a positive left to right pattern of enrichment, with more enrichment seen in the cancer group along with the HKNR group. Similar pattern is being observed with TCG head and neck cancer dataset, where a widespread is being seen in the pre-malignant patterns. And finally, the partial EMT profiles, which is a special profile in head and neck cancer and has also been associated with an aggressive state, can also be seen to be enriched in our pre-malignant lesions as well as cancer group. This characterization helps us understand that the overall pattern of enrichment for malignant transforming as well as metastasis signatures is being enriched in our PML as well as our cancer group. To move on, we applied UMAP reduction, dimensionality reduction analysis on our data set with considering the top 2nd through 10 principal components that were actually derived from top 500 highly variable genes in the data set. So as we could see in this plot, the control group is being secluded in the lower half of the UMAP plot, whereas the cancer group is on the top right corner. And the HKNR as well as dysplasia span across both these groups, forming a gradient. So to cluster these samples into highly formed clusters, what we can do is we can apply a probabilistic approach to get the number of clusters in a data set, which came up to be 3. And applying this approach, we see that this cluster is predominantly the control group with more than 50% of the samples. This one is predominantly HKNR, but dysplasia and cancer secluded towards the top right corner, showing much higher chi-square score. So this UMAP plot gives us an idea that the transcriptional profile shows considerable heterogeneity. And we also did differential expression analysis to understand the gene level patterns between these groups. So we did this in a pairwise approach that led us to a 3-part classification. First, we compared the non-control group, that would be the PML as well as the cancer, with respect to control. Then we did PML to cancer and then in between PML. So the results, what I'm showing you here, are from the gene signatures which come up as significant. For example, the upregulated signatures was obtained with a cutoff log fold change of 1.5 with a FDR corrected value of 0.05 and the downregulated of log fold change of -1.5 and an FDR of 0.05. So we see that when we apply the hallmark compendium using a hypergeometric type of enrichment analysis, we see most of the pro and anti-inflammatory pathways being enriched when compared with the control group. For example, the cancer shows here that it's been upregulated in epithelial to mesenchymal transition as well as interferon gamma and so on. Similar pattern is being observed with the dysplastic group, and then when we compare the PMLs with the cancer group, they were downregulated, whereas the cancer group was upregulated in this case. So the comparisons also showed similar patterns when you consider the reactome compendium, which is more on the microbial as well as other extracellular matrix kind of compositions. So the differential signatures were tested on this compendium, and we see that the PMLs as well as cancer is being enriched for some of these patterns which come up as extracellular matrix composition that could arise from the degradation of extracellular matrix as well as the activation of matrix metalloproteinases. Similar pattern was seen with the innate immune system, and a special profile or mechanism called Fc gamma receptors phagocytosis was also enriched in our cancer group as well as the pre-malignant group when you compare the HKNR with the dysplastic group. So this gives us an indication that the microbial activity is being influenced on the activation of some of these pro and anti-inflammatory pathways. So since we did total transcriptomics, we can also get the relative abundance of some of the microbial communities here, and this slide shows the composition of the microbial abundance stratified by different OTUs. For example, you see at the phylum level, the diversity is being pretty much similar across the groups, and the similar patterns was observed with the genus as well as species level, with an indication of some of the disease-causing microbes such as Fusobacterium being more abundant in the cancer group, as we could see here in this blue plot. And for our downstream analysis, we considered the genus level rather than the species, because some of the species in our data set were unidentified. So one could also look at the richness of the microbial diversity in each of these histopathological groups. So this was done by first identifying the microbial changes in samples, so that can be identified as an alpha diversity. And here we can see that the HKNR group is, as well as the cancer group, is highly rich compared to the control group. And then we can also see the beta diversity, which is comparing the samples between groups, and we see the dissimilarity of the cancer samples having different microbial communities compared to other histopathological groups here. And then we can look at the relative abundance of some of the disease-causing microbes, which has shown to be rich in our samples. Like, for example, Fusobacterium is being associated with several cancers, including oral cancer. And then you have Neisseria, which is common among alcohol drinkers. And then Pravitol and Chavinella has pathogenic properties and also been associated with periodontal diseases. And then you have Candida, which causes fungal infection. So this pattern gives us an idea that some of the disease-causing microbes have an influence on some of the cancer or pro-inflammatory pathways. And this leads us to characterize the microbes along with the gene level information to build a crosstalk model. So for this, what we can do, we can understand this, for example, if there is no microbial clearance or there is abundance of certain microbes that can lead to the activation of chemokines and cytokines along with pro-inflammatory pathways such as interferon gamma, TNF-alpha, and so on, which I've shown previously. So to study the host microbial crosstalk, we employed the microbe set enrichment analysis method that takes into consideration a reference microbe gene interaction compendium, which has been collected by aggregating results from published literature. And it also needs a signature of microbes to check for the enrichment and gives out a combination score that can help us see if the microbe has been positively associated with the gene or not. So for this, we considered the microbes. For example, this is with the cancer group, so you would do differential analysis comparing cancer to the control group, and the microbes which come as differentially abundant can be used to identify or check for enrichment of some of these genes, and that is what we see here. The red edge indicates a positive association or the combination score that spits out of MSEA algorithm, and we see, for example, Fusobacterium as well as Campylobacterium with other has been associated with IFI16, which is an interferon gamma gene, along with IL6, and these associations of pathways also comes up in our host, for example, the human analysis what I showed previously, and these can be used to build a model that helps us identify the interactions between microbes and genes. Similarly, we applied the PML group in a similar fashion, and we see very similar patterns of enrichment as we could see with the cancer group. So this helps us connect these genes to pathways that are pro-inflammatory and also understands the microbial genera's influence on the activation of these pathways that drive them into malignancy and transformation into tumorigenesis stage. So the take-home message is that the PMLs are enriched for malignant transforming and metastasis signature, and it has been shown to be considerably heterogeneous, and then the differential analysis showed activation of pro and anti-inflammatory pathways along with an immune activity, and then certain microbial genera which has been considered has shown to be associated with the activation of immune pathways that promote malignancy. With that, I would like to thank our collaborators and our lab for helping in constant feedback as well as ideas that went onto this presentation. Thank you.